So now moving on, we're finally going to make some DNA. So we're going to introduce our major protein that synthesizes DNA, and that's DNA polymerase 3. So DNA polymerase 3 and its subunit, this sliding clamp called pCNA, work together to read the template strand. And it's important to note that it's reading this template strand here, shown in blue, in the 3' to 5' direction. And then it's synthesizing the new DNA strand in the 5' to 3' direction. And that's because these two strands will have to be anti-parallel in this semi-conservative mode of replication. So once again, it reads the template in the 3' to 5' direction, and it synthesizes new DNA complementary to that template by inserting nucleotides in the 5' to 3' direction. So you can see here it's moved along and it's moving towards the replication fork, inserting these nucleotides. So this strand where synthesis is moving towards the replication fork is known as the leading DNA strand. We'll get to why that is a little bit later, but essentially that means that the new DNA is being synthesized in the 5' to 3' direction in the same direction that the replication fork is progressing. So the fork is opening in this direction, moving towards the left, and the polymerase is synthesizing the new DNA towards the left as well. So I want to take just a minute to talk about the enzymatic reaction that DNA polymerases catalyze. You should know already that combining nucleotides, the monomers of DNA, to form these DNA polymers, these polynucleotides, is performed by dehydration synthesis reactions. So these are reactions that produce water, and these are classically endergonic reactions. However, we have to have some way to make this favorable because this process has to happen under normal cellular conditions. So how DNA polymerase allows this process to be favorable is it uses a different kind of substrate. So these are the substrates for DNA polymerases. They're called nucleoside triphosphates, or DNTPs. The important thing to note here is that instead of just having a single phosphate, like we talked about a nucleotide, they actually have 3 phosphates attached to this nucleotide. And that's going to be a major source of energy for this reaction, because when this complementary nucleotide is added to form its base pair, and then we're going to form a phosphodiester bond between these two nucleotides, 2 of these phosphates are removed. This 2-phosphate group is known as pyrophosphate, and that releases a lot of energy. In fact, release of pyrophosphate provides sufficient energy to make DNA synthesis an exergonic reaction. So release of pyrophosphate from DNTPs is capable of driving what should be an endergonic dehydration synthesis reaction, making it exergonic. So here you can see we've now formed our new phosphodiester bond, and our new strand is complementary to the old strand. So we have A base pairing with T, C base pairing with G, and so on and so forth. Now I want to consider, we talked about the leading strand, where this DNA polymerase is moving towards the replication fork. But what's happening on the other strand? So on this strand, remember we already talked about that our primase and our DNA polymerase can only synthesize DNA in the 5' to 3' direction, and they can only read the template in the 3' to 5' direction. So something different has to happen on this other strand, and it's known as the lagging strand. So because DNA polymerase can only synthesize DNA in the 5' to 3' direction, and because these two strands are anti-parallel, this means that essentially as this fork opens, DNA synthesis has to start over again and again and again. A new primase has to come in and add a primer, and then DNA polymerase III can extend from that primer. And as this opens a little more, a new primase will come in, add a primer that then DNA polymerase can extend from. But here, DNA synthesis is progressing away from the direction that the fork is moving. So this is called the lagging strand because those polymerases and primases have to keep starting over again and again and again. Now, just to remind you, on the lagging strand, DNA synthesis is moving opposite the direction that the fork is progressing. So the fork is opening towards the left, but DNA synthesis is moving towards the right on the lagging strand. Okay, so now we've still got some problems with this DNA strand because, one, we have these pieces of RNA that are still a part of it, and we have these gaps where DNA polymerase III extended until it reached the primer. So now we need to introduce a few new enzymes. So here we have DNA polymerase I. So DNA polymerase I is a different DNA polymerase, and its role is first to remove the RNA nucleotides, so to remove those RNA primers, and it does it using a very specific activity of the enzyme. This is called the 5' to 3' exonuclease activity. So exonuclease means to remove nucleotides. So it removes RNA nucleotides in the 5' to 3' direction, but then it replaces these RNA nucleotides with DNA also moving in the 5' to 3' direction, because like DNA polymerase III, it has a 5' to 3' DNA synthesis activity. So that's the role of DNA polymerase I. And then our last enzyme is DNA ligase. DNA ligase is an enzyme that uses energy from ATP to seal the nicks in both the leading and the lagging DNA strands. So essentially it makes phosphodiester bonds. Any nicks where these strands aren't connected to one another are sealed by this DNA ligase enzyme using energy from ATP. So just to review, we have several major enzymes to consider. So helicase unwound the DNA helix by breaking those bonds between the base pairs. Single-stranded binding proteins stabilized that single-stranded DNA, protected it, kept it from being degraded by nucleases, and kept it from closing back together, reforming base pairs. Our topoisomerases relieved that over-twisting supercoiling that happens ahead of the replication fork. Primase was an enzyme that added those RNA primers, allowing DNA polymerase to then extend from those RNA primers. DNA polymerases, unlike primase, cannot do de novo synthesis. However, they could extend from those pre-assembled RNA primers. So we have DNA polymerase III that makes most of the DNA, and then DNA polymerase I, whose role is predominantly to replace the RNA primers. So it removes the RNA primers using its exonuclease activity and replaces them using its DNA synthesis activity. And then finally, DNA ligase seals any nicks left over after the RNA primers have been replaced with DNA by making phosphodiester bonds using energy from ATP. So you should know each one of these enzymes and be able to describe their functions. You should also be able to draw a replication bubble, and you'll get some practice with that using the activity that is associated with this lecture. So just to review, I want you to answer this for yourself. So which direction does DNA polymerase read the template strand? So not the direction it synthesizes, the direction it reads. So take a minute and think about it. Did you write your answer down? So if you said 3' to 5', You are correct. So DNA polymerase reads the template strand 3' to 5', and it synthesizes the new DNA strand 5' to 3' on both the leading strand and on the lagging strand.